Welcome to the Proteomics in Proximity podcast, where your co-hosts Dale Yuzuki, Cindy Lawley and Sarantis Chlamydas from Olink Proteomics talk about the intersection of proteomics with genomics for drug target discovery, the application of proteomics to reveal disease biomarkers, and current trends in using proteomics to unlock biological mechanisms. Here we have your hosts, Dale, Cindy and Sarantis.
Welcome to Proteomics in Proximity. I'm Dale Yuzuki, your host, along with Cindy Lawley and Sarantis Chlamydas. And here we have a special treat today. Sarantis?
Yeah, thank you, Dale. I'm really happy to be back from holidays and then having our first host, Maria. And Maria is a student, she's a PhD candidate, she's a PhD student in SciLifeLab, and she had recently a great paper. She was the first out of a great paper that we will discuss a little bit later about this more in details. Maria started her studies in Barcelona, in Autonomous University of Barcelona. Then she moved to Stockholm and she gained the master's degree, joined the Protein Atlas, the Human Protein Atlas project in 2020. And since 2022, she's part of Mathiasson Ulen Lab, and she's working in an amazing project around proteomics in different diseases and cancer also, including cancer, try to identify signatures, protein signatures. Maria's background is around, of course, proteomics, transcriptomics, and she's also bioinformatician and wet lab scientist. That means she has a complete package of a full, experience and expertise in the field, also in the field of multiomics. That's something I'm really interested also to hear. And I will start my question, actually, Maria. I mean, we are really happy to have you. And it's always a pleasure to discuss with you. And I would like to know a little bit more, how will you see your transition from transcriptomics to proteomics? And how do you see the match between these two omics approaches, actually, from your perspective?
Yeah, thank you, Sarantis. It's really great to be here today. So I think for me it was a very smooth transition, basically because at the Human Protein Atlas, that's been kind of a very connected thing. It's never been like transcriptomics is one thing and then proteomics is something completely independent. It's always been quite linked. And that's because, of course, Human Protein Atlas has been about understanding proteins. But we have not always been able to just look at them right away in such a scale, right? Now with Olink, it seems like that has been there forever. But in the very early days, transcriptomics was really, really useful tool to look at the proteins, indirectly, of course. But it's still, we learned a lot, I think. And it's a resource that it's still used to this date in many, many groups. So I was really lucky to work with the transcriptomics first, I think, so that I could understand, I sometimes kind of say that that's the simple part where everything is clean and you don't have so much noise of post-translational modifications and so on. So I think it's more of the straightforward tissue transcriptomics analysis. But now, of course, it's really great to go to more of plasma proteomics, which is kind of the other extreme.
To ask you a little bit about the transcriptomics piece, it was there at Science for Life Laboratories, right? And was the interest there in transcriptomics around a cancer indication, or was it just across tissues?
Yeah, so the very early transcriptomics, that was published around 2015 in Science, and that was healthy tissue. So that was kind of a really big study across different tissues, tried to just characterize which proteins are specific. And that was kind of the flagship of the Protein Atlas, tried to just see what's there. But then, of course, there's been other more integrating with GTEx data and trying to understand, of course, a little bit more in disease.
And so this transcriptomic data set was from the tissues from Science for Life Laboratories, right? It like you mentioned, there are other projects that are characterizing tissues like GTEx. What made the Science for Life effort unique or different?
So nowadays at the Human Protein Atlas, we have both data from HPA and GTEx integrated into the database. When I say HPA, I mean Human Protein Atlas. It's just a little bit too long. So our own samples, for example, we have a really detailed brain collection of samples. So it's not only brain, but we have I think it's tens of different regions in the brain. So for example, we have a ton of detail on that stage that you can't really have with GTEx data. So it was more trying to complement, and also, of course, if you have two different independent sources of data that agree on the same transcriptome levels, that's of course very reassuring. So that's why we try to combine both in the same database.
Amazing. So hey Maria, this is my first chance to ask a question, and I'm actually gonna click back to ask about your interest in getting into science. So and usually we say you don't have to start with elementary school, but if you want to, I mean, whatever, sparked this interest in you, I'd love to hear about that. And then what nurtured that interest to end up in Matthias's lab? I mean, what a remarkable place to be.
Yeah, no, thank you for your question. It's of course always difficult to know when it all started, but I think you will probably click with this, that biology is just fascinating. Like, you see people getting old, people getting sick. I don't know, I see the effect of epigenetics as well. I have a twin sister myself, so I was always like surrounded by what I thought was biology. And then I thought studying genetics, which was my bachelor's, that would allow me to see why this is happening, why we're getting old, why we're getting different, why we're getting sick. So I think that was for me what raised my interest in science. But I think also back then I didn't know how science really looked like. So I didn't know about scientific research and so on. It was more of a dream, right? Like, let's work on something fun. But then of course about Matthias' group, that was when I moved here to Sweden. Of course I had heard about the protein atlas before. We used it in our bachelor's and during the master's, but then I think moving to Sweden was really putting me very close to his group and to his research. So you learn a lot during the master's about the database, and I also got to know people in the lab. So that's really how I ended up there.
And you moved through, it sounds like you committed to doing a master's first, and then that evolved into doing the PhD. How did how did they keep the carrot in front of you that you kept moving through that? Because it's, I love the stories behind making these decisions. I think students are hungry for other people's stories about this.
Yeah, I think for me it was moving here to SciLifeLab, or starting my master's here. I had not connected so much with research and like academic life at my previous university. Not because of the university, I think it was more of the like how bachelors are structured in Spain and in many other countries. You don't really get to feel how it is to be a researcher. But then I moved here to Sweden, and I started this program. And they had a really nice mentorship initiative where you could get a PhD student as a mentor. And for me, that was really what changed my perspective. I was paired with Max Carlson, a PhD student here at Matthias Uhlens. And that's really when I saw, okay, this is what I want to do. This is what I want to be.
That's amazing. I love that story, the mentorship, and that the fact that they structured that with the mentorship. That's amazing. So then this publication that Sarantis hinted at and that Dale talked about in the intro, this is essentially a pretty remarkable set of data looking at over 1,400 patients that had across 12 different cancers, and about 14. This was our Explore 1536 Olink tool, and so I think 1463 proteins were included in this study. And you were tell us a little bit about this study, because it's a remarkable paper.
Yeah, no, for me, I was already working at this group when they started like the planning and everything was being run. So of course, hearing about, the total number of samples for the first phase was 10,000. So this is only the cancers that we're talking about. So I was hearing about this and I was, this is unbelievable. I had already worked with Olink data, but it was only a few panels. So also moving to Explore, I was really, really interested. And the data was delivered at the very early 2022, or maybe even December 2021. And that was exactly when I started my PhD. So you can imagine how it felt like, well, this is First of all, a Christmas present and then a welcoming present. So yeah, that felt really, really exciting. Also because we didn't know anything back then. Like, is there going to be any signal in this 10,000 samples? So that was really exciting times.
And you already had pretty strong experience in bioinformatics, right? So I imagine it really was like
Luckily, yes. Yeah. Otherwise, it would have been a bit scary maybe to get all this.
Jump right into the deep end of the pool.
That's right. That's right. And so you were the 10,000 samples. Is that including the UK Biobank? Are those all within the UK Biobank? You also in this study used the Scapies Wellness cohort that's also under the auspices of the SciLifeLab, as I understand it. Is that right?
Yes, so these 10,000 samples, that included UK Biobank, which is this Swedish initiative which collects samples from many different cancers. And that's the study that is now published. So that's around 1,500 samples in total. And then we also had other cohorts, like you mentioned the Scapies, you mentioned wellness. So this is other Swedish and also non-Swedish cohorts from other different diseases. We also have infectious data, infectious disease, autoimmune disease from Karolinska Institute. So it's really a lot. And of course, we had to break it down. And that's kind of the first published study. But we have a lot of lines of research open right now.
Can I ask you some technical, and I'm sorry if it's a very naive question. If I understand correct, the controls were for a wellness cohort, for a different cohort, right, in a way. Do you think that, is it easy, or how easy it is to just have a control for a different cohort in order to have, let's say, a well-studied project? From your perspective.
Yeah, so ideally you would always run case control, right? Like you would have, for every single disease and every single cohort, you would have match controls. That's usually possible, but then, of course, if you run 10,000 samples, it's a real loss if you just need to spend 50% of that on controls. So we've been trying to include different sets of healthy cohorts. So not only wellness, but we also have, for example, we talked about the Scapies. Then we have also healthy individuals, also from Turkey. So just try to have many different ones, so we can have a look at the pre-analytical variation, and we can see that everything makes sense across cohorts. But of course, that's always a limitation, right? In these big studies, that you can't. You have to live with that, with maybe a couple healthy cohorts, but not much per disease.
No, and I think also the fact that you have different type of cancers is a kind of internal control in the way, right? Because you expect to see this classification in a specific type of cancer rather than the other. That's a kind of, there's specific effects happening in one like the other. And also it's a kind of control. Therefore, this makes the study really, really, really amazing. Can you give a comment? Because when I start reading a little bit, and can you give a comment about the lung and colorectal cancer? There's something common between these. I haven't heard about. What is your feeling, and what do you think is happening in this type of cancers, having so much commonalities rather than differences?
And just for context, I'll just say those were 2 of the 12 cancers for which the team characterized the ability to differentiate among these cancers within this study. So that was the primary focus and the exciting result, which allowed for leveraging machine learning in characterizing those signatures. So I think these particular cancers were a little challenging, perhaps, but also an opportunity for advancing our understanding. So yeah, Maria, please.
I'm happy you asked about that, because as a bioinformatician without a very strong medical background, that was my first question, like, what is this? So that shout out to our doctors that we are very lucky to have and call in anytime we have these kind of questions. So we had very strong discussions about this, because it's kind of the biggest overlap that we saw. So there is an overlap between the immune cancers, so they have some shared signals, but also this lung and colorectal. And I was really wondering, like, why could this be? And after meeting with them, of course, we would have to look farther into this, probably get more samples with more detail, histology and so on. But their guess is that we are looking at 2 cancers that are mostly adenocarcinomas, And it's probably a common signature in the pathway in the development of this type of cancer. So it was mostly apoptotic signal, and I think also like cellular stress, which is quite a general thing, but it makes sense that we don't really see that in, for example, immune cell cancers. Like it's for them, it was quite obvious, but I had exactly the same reaction you had now. Like, why would that be? So yeah, that's a nice thing about collaborating with the doctors.
And I think this ability to it's almost like I picture it as a Venn diagram of these, what seem like quite disparate diseases. The pathways that are overlapping in these diseases, I think, are giving us a sense of that mechanistic biology. So I think you're turning yourself into someone who understands that, as well as the bioinformatics, like Sarantis said, quite a full package of someone who can work with these data and their complexity.
I mean, on that note on data complexity, if I understand correctly, it was in review for many, many months, right? I think the preprint showed up maybe December, January, and it wasn't published until late July. Can you comment on that in terms of the review process, in terms of what you can share? Was it because the data with the machine learning, right, in terms of those algorithms, was that where a lot of the review work entailed?
Yeah, so I think you understood a little bit of the complexity. So it's really huge amount of data, and when you're dealing with this kind of data and machine learning, it's a little bit dangerous sometimes. So it's kind of you need machine learning to understand it, but then you should be skeptical about it. So I understand 100% why it took maybe a bit longer than expected, because you need to make sure that the pipeline is rigorous and that there's enough controls of, that it's not noise that you're capturing. Of course, that we will never know until we replicate or validate our findings in different cohorts. But yeah, to be sure as much as possible that this is a really well-structured study. And I'm happy that we went through this revision so that everything is in place.
Yeah, when I saw some of the first reaction on social media, namely over LinkedIn, it was interesting, right? One of the, not criticisms, or may have been a slight criticism, that these were post-diagnostic samples. By that, I mean, this was taken untreated individuals at the time of diagnosis, these 1,400 samples. What can you say about that in terms of, could you have gotten pre-diagnostic samples or what have you for an early detection?
I really understand. And I mean, I would call it also a limitation, right? I mean, it's, of course, you need like many different kinds of studies. I think this one is very interesting. But then, of course, knowing that these proteins are also up before diagnostics, that's of course really, really interesting. But in our case, we had access to this biobank, so we couldn't just start now, and we would have to wait 10 years, right, to get all this data. So we still think it's valuable to know what happens at diagnostics, at time of diagnosis. We also, for example, we had early stage and late stage samples, so we also think it's relevant to see that these proteins are also up in early stage. And I can comment maybe that now with all this UK Biobank new data that has been released, there was, for example, a preprint, I think it was last week, also on different kinds of cancers. And I was, for example, very happy to see that a lot of our proteins that we found upregulated, they also see 7 years prior to diagnostic. So I think it's valuable and complementary maybe. So we don't have to wait so many years to see when we already have samples. But then, of course, if we also have that data, that's definitely great.
I think that was one of the most fascinating aspects of your paper, in that you found in your signatures across the 12 cancer types some well-known markers for well-known cancers have been researched for extensively, and then other new markers that imply new mechanisms. And what can you comment on that?
Yeah, for me, that was the most fascinating because it's great when you find the top marker and that's already been found, because then you're sure that you're doing things right. Exactly. But if everything was known, then why am I even working with this, right? So it's, I think it's good to have a combination of really well-known markers, unknown, and maybe some of my favorites are those that they have been described somewhere in 2015 in a random paper, but no one has ever looked at those, and they have been forgotten. And then you find it, and you dig in the literature, and you realize, wow, this was linked to myeloma many years ago, and I'm seeing it again. These are my favorites.
So here it is. You heard it here first from a bioinformaticist that she has favorite markers.
Big surprise. I do want to talk, it's just mention that preprint. I'm so happy you talked about it. I think that was Ruth Travis and Karl Smith-Byrne and Josh. And I just have a little tiny story. When we were at ESHG, Joshua came up to our booth and he told us how excited he was about the UKB data being available, that he's been sifting through it. And he was the one, Dale and Sarantis, I think I told you this story before, who just kept telling me, I can't tell you anything about it because we're coming out with a preprint, but holy crap, holy crap, holy crap, holy crap. He was so excited about mining those data. And so if he's a representative of the kind of excitement that a resource like that can generate, I think we're going to have some really fun conversations in the next few months about what people are seeing there. So it's thank you for highlighting that preprint.
Truly pioneering work, right? There is only one first. And like you mentioned, you're looking at some random paper in 2015 that had some association with one of those 12 cancer types. And all of a sudden it pops up, right? Yeah. As being an important. Oh, I thought that was very interesting too in the analysis where you actually look at the weightings and its influence on the overall power, right? In terms of that signature goes.
Yeah. And I think something that is also, for me, a take home from this study, and we probably haven't talked about it yet, is that, not all cancers have like the same amount of proteins that are important, right? So you can have, maybe you need 20 proteins to characterize a cancer, or you might need 200. And I think that was not so evident for all of us a few years ago. Like we were hoping to find one protein per disease.
Oh, the smoking gun. We want the smoking gun.
Yeah, the one single biomarker, right?
It's so much easier to get clinical utility past the FDA, and they love having one single test, one single HDL, LDL, these are, but it turns out they're complex.
That would be ideal.
Mm-hmm.
But the body's so good at repurposing all these proteins for all these different tissues. I think it's a good lesson. I will say I'm just chuffed. I love this figure, and having Mateus talk through this figure, which really characterizes some of the signatures that are showing up stronger in some cancers than others. And then the connective diagram that you have. Sorry, we don't use slides on this podcast, but I will say that the images are gorgeous that allow us to just talk about what you've done and help folks understand the significance of the approach across multiple different diseases, right? So, and then I think you capture it well around the importance of early cancer detection and these tools around genetic detection of cell-free DNA, for example, in blood. They'd suffer from false positives, and that's a big concern that folks will be given a potential diagnosis that isn't real because we want to make sure we capture some of these cancers in early stages that are real. And so being able to add value to that approach, do you have any thoughts about how this might complement that, how it might be leveraged in combination with some of these innovative new approaches?
Yeah, no, that's a very, very relevant and a very relevant comment. And we talk about it a lot here because, when we see the false positive rates, and it doesn't look so bad when you look at the numbers, but then if you think about a population scale, then that would be a disaster, right? If you could call in so many patients.
And an individual patient experience too, right? How that would feel to an individual exactly what you say.
Yeah, and what we've discussed before is that it's of course really nice with cancers that you have like external means of getting a validation that the person has the cancer. So instead of just running the blood test and saying, oh yeah, you're diagnosed with the cervical cancer, if you would have a way to verify that, like for example mammography or some other test, then it's kind of easy to convince the population that this is only a screening, that it's not that sensitive, and then if you get the false alarm, then you of course get sent back home when you get the negative second test.
Yeah.
But when I think about for example cell-free DNA and other ways of finding cancer that are very promising, I think that if we could maybe combine cell-free DNA testing with proteomics in plasma, that would probably filter out some of the false positives. Some people that would have maybe DNA signal but are not so strong on the proteomic side, or the other way around. So I think it would be at least 2 layers where it's more difficult to be a false positive, or on the other hand, a false negative.
Yeah.
So I think maybe
multi-omics, multimodal, those words.
Yeah. What a lot of people found out at AACR in Orlando, right, was the readout for the phase 3 clinical trial for the Gallery Grail test. And the fact is, it just replicated the phase 2 and phase 1 pivotal trial or clinical trials, in that there is about a 50% false positive rate, even with 99% specificity, just because the prevalence of cancer is about 1% in the general population, right, of normal risk. And that is a problem, right? But then to think, and therefore, right, the harms that happen where people are saying, well, we have the signal, we're not sure, right, if you have cancer or not. You have to look for regular diagnosis, and naturally for colorectal or for lung adenocarcinoma, it's all very straightforward in terms of diagnosing those. But the Gallery test is looking at 48 other cancer types, and some of them really do not have gold standard diagnostic methods. And so patients can be sent, right? You might say, well, it must be terrible to be that person. Yeah, right? Where they're told you might have cancer and we can't really find it using our best diagnostic tools, a real genuine harms, right, to individuals. And yet at the same time, the value of early detection, right? The incredible power to prevent death, right? To catch it in stage 1.
Yeah.
If you know about this false positive rate and you can't validate that is indeed a cancer, then you're also not going to treat it, I guess. So,
right.
Yeah.
I mean, for me, I think that in the ideal case scenario, you would just be able to sample everyone longitudinally through their lives. And I think then it would be really easy. I'm saying easy, but it would be easier to find differences in your proteome and like actual pathological states. But now, I mean, you're sampling a person, comparing it to some threshold, some other data that has been collected. There's going to be a lot of noise.
Yeah.
But of course, that's daydreaming. Like, what would I like to have?
Yeah, for sure. And next steps, right? Sort of what's in the future. In this study, you were able to do sort of a 70-30 split to be able to train your machine learning around a set of the samples and then validate with a subset of those samples. And I think your areas under the curve for across all cancers were like 0.8 to 1, but really, really high in AML, CLL, and myeloma, as I wrote down in my notes. I think that next steps are for validation. Anything you can say about what you all are planning or what you hope others will do to help validate this so that we understand its power?
Yeah, so, I mean, ideally, as I said, it would be nice to just work with these cancers for many, many years and get additional cohorts and validate all these findings. But our plan for now was to share this data, share all the protein levels in the database. So for example, now the preprint that we just referred to, of course, they found exactly the same proteins in myeloma. And that was really, okay, we haven't validated ourselves, but these people have found it in Sweden, right? Yeah. So it's not only us to maybe validate these findings, but we also hope that other research groups will refer to this and realize that they have the same thing in their own data, maybe even with other methods. So it was more to share a list and to share our results and hope that also other people will do the same.
Yeah, and I think this ability to, because I can imagine, sort of these 3 to 12 to however many proteins are needed for each cancer, those tests. But then you did this beautiful thing, which is combine the proteins that were across all of these into an 83 protein panel that was, I think, really. I'll let you describe that. I think it's a great part of the paper that I appreciated and wanted to highlight.
Yeah, no, so of course, I mean, the most important is to learn about the proteins that are important for a disease, or that was the aim. But what we wanted to see also is if we can go down. So instead of looking at 1,400 proteins, what happens if we just pick what we think are the most representative? Do we still get good classifications? Are we still able to separate all these samples into their specific cancer of origin? And I think, I mean, of course, this is only the first study. We haven't validated, but it looks quite nice that only using 80 proteins instead of 1,000, you can really guess what that sample is. And I mean, in the future, that I think would be a really nice way to look at diseases, to just have a list of markers for not only cancers, but maybe other diseases that could be related, and then just try to see where your sample falls. So based on these markers, you're most likely cervix cancer patient.
Yeah, I have actually 2 questions. The first question is how do you see? For sure, there are other databases with other disease, right? How would you see these proteins, 83, correlate with other disease? I mean, how unique, because you haven't done this correlation, how unique you just check between cancer, but how unique is compared to other immunological disorders, for example?
Yeah, so, I mean, for that, you will have to wait a little bit. I mean, as I said, we have many, many different diseases now. I think
Super.
We have around 88 in phase 1,
Super.
And we are planning for a second phase. So, yeah, expect us to write about this.
Super.
I think it's also been highlighted in other pieces of work that there is a few proteins that are found kind of everywhere. I think GDF15 is one of them. It's some proteins that are just up in also immune proteins. They are probably, inflammation is everywhere. But there's also quite many that are very, very specific. So I'm always reading Olink papers because it's really fun to see, okay, this protein that popped up for colorectal is also maybe related to HIV. I don't know. Wow. So it's quite nice.
That's just so cool. And expanding the protein panels, right? We've just announced the expansion to 5,300 proteins, which, sorry, go ahead, Sarantis. You had another question.
I was just to mention this. Yeah. No, perfect, Cindy.
We think the same, I think.
Just thinking about what you just said in terms of proteins that are common to disease, that goes beyond just cancer or just autoimmune or just infectious, and to think, wow, to have a certain set of markers that, there's something going on here, or you better get this checked out. That is wild.
I guess you could start monitoring these more general markers, since it is probably expected that more specific markers will pop up later on in the disease. And then once you know something is wrong, you could go deeper and run another panel maybe. And I just want to comment on this new release of the Explorer HD, do you call it?
Yes, that's right.
I think that's really exciting, at least from my side, because when you work in biomarker discovery, I don't think a lot of people are used to this really high number of targets. And for me, already 1,500, I felt like that was amazing, right? Like, eventually you start getting to know some proteins by heart, but it's still a lot of proteins to look at. But now, I mean, of course you had 3,000 in between, but for me, going from 1,500 to more than 5,000 proteins that we will have on the next phase, I'm really looking forward for that. That's going to be a really nice, it's a really nice list.
It's a good chance to mention that just this week we've integrated that entire list into our Insight app. So this is a freely available software that has a web-based interface at insight.olink.com. And you can browse pathways, see how many of the total proteins in those pathways we have on that HT and our other panels. I'm in there all the time. I was surprised that I'm a super user of it. So clearly it's underutilized because so it's relatively a new set of tools that we've provided in the last year, really. And the data stories in there include the Cancer Protein Atlas that we're talking about here today. So folks can browse some of the results that you found in that exciting study, which, by the way, Nature Communications, you're a PhD student. Look at you, Maria. Amazing.
It is amazing. On that note, Maria, right? There is an idea for a disease protein atlas. Is that correct? Not just cancer, but other diseases. What can you comment on that?
Yeah, so right now we have published these stories, so that's the data that's available now on the atlas. It's still called Human Disease Blood Atlas, although now it's more like a cancer atlas. But the idea is we're going to keep on releasing data. So I mentioned phase 1 that has infectious diseases, it has autoimmune disease, and many others. That's going to be soon, relatively soon, part of the database. So. And then, of course, we have plans to expand to a phase 2 and have more targets. So that's also going to be part of the atlas and hopefully the Olink Insight as well, so that it can reach as many researchers as possible.
That's an amazing resource. Doesn't Matthias talk about how many millions of hits and how many thousands of pages of data freely available?
Yeah, I mean, it's really a unique effort, I'd say. And also, I kind of really like the history of like focusing on proteins and now going a little bit more. We had this health study and now we have this disease study. So I think it's quite a natural course of how you should look at biology.
Right, from the healthy to the disease and look at those differences.
Maria, talking about a little bit biomarkers, and I know that the scope of the project is around identifying biomarkers for prognosis, but for sure among these biomarkers there will be targets, right, for drugs. I mean, are you considering to take a look more to the drug development perspective? I'm sure that you are, but have you found some really cool new targets, for example, that pharma companies could take advantage on?
And do you need
if you want, I mean, if you want to share, or it's too early, it's up to you.
Do you need more cohorts to validate those drug targets in? Yeah, both of those. Great questions.
Yeah, no, so I think you're both on the right path. Like we, of course, understand how valuable this data is. And we are, of course, starting to see some proteins that look very interesting. But as you both mentioned, it's early in the sense that you need lots of validation. So we want to be careful with that. We are, of course, look at different markers. We have an eye on them, but it takes a while before you can say anything about the disease.
But crowdsourcing the data like this, making it publicly available, allowing different folks with different ideas about how to analyze these data, have them debate over social media, as Dale was alluding to, really moves things along quickly. In my experience, I think the genetic history is a great testament to that. I think we're seeing that now too. So I'm excited that you're making these data publicly available, and Matthias's commitment to that throughout his career.
Yeah, for me that's a really important thing. That of course it's great to move forward in your own science. But what you said, for example, about drug target development, maybe some people doing their own research, they will see this resource, they will find, okay, this is really a good target, and that will inspire them. So I think Matthias' spirit has always been to share and to inspire, and I think that's a great way to do science. At least I'm very happy I'm here.
On that
I think that's an important point, right? Because wasn't there a decision made on the analysis to focus in on the upregulated proteins in the disease samples?
Yes. And that's also been a long debate, of course, because, typically upregulated proteins are very interesting because it's like it's kind of easy to detect them when they are high compared to a healthy population, right? And also sometimes, like for example, we saw with AML, with leukemia, that you had this really good marker that was upregulated and this really good marker that was downregulated. And you would think maybe let's take both, but many times they are just correlated because one is the receptor of the other, right? So in a way, sometimes up and down regulated proteins, they are still in the same pathway. So maybe it's not that interesting to focus on both at the same time, since they are leading to the same story.
And
so
then
in our case, yeah.
Well, the other piece, though, is that if you can modulate the upregulatedness of the disease protein, right, that actually is a drug target, right, where drugs are really good at lowering proteins. There are only a handful of drugs that upregulate proteins.
Yeah, I guess when we talk about it being more classical from the clinical perspective, it's related to exactly that it's kind of easier to suppress than it is maybe to just somehow make this protein be produced or provided.
Yeah, I've never thought about that. That's a really interesting point. Yeah, thanks.
Yeah, so therefore, right, we have drug targets that you need to lower in order to lower whatever incidence or whatever mechanism that's driving the cancers. And what's, I think, fascinating about cancer biology, after all we've learned in the cancer genomics and transcriptomics, here it is, you're looking at protein level now. Maybe we're circling back all the way to the beginning of the conversation, right? You'd started with transcriptome, and wow, once you look at the proteome, everything is, right, illuminated in a new way.
Yeah, that's the real thing, right? I mean, we are proteins. So it's really, really nice to be able now to look at proteins directly. And see, also in blood, like you don't even have to go to your pancreas to study pancreatic cancer. You can hopefully just look at your blood. I
never thought of that before. I am protein.
Yeah.
We are protein.
I think
that's because you haven't talked to Matthias.
Hard to get on his schedule, but we've got a surrogate for Matthias right here. So I think this will be our new mantra here. I think you've made an impact, Maria.
Every one of us is protein.
That's right.
What a thought. Well, Maria, thank you for your generosity with your time today. We really enjoyed this conversation.
Yeah.
Thank you for inviting me. It's been really great to discuss with you.
Yeah. And then not to forget to thank all of these patients that consented to allow, now and in the future, all the work that you and others do. I mean, what a wonderful, generous act. And without it, we wouldn't be where we are today. So
for sure.
Possible.
Amazing.
All right. Thank you.
Thank you.
Thanks so much.
Thank you.
Thank you for listening to the Proteomics in Proximity podcast, brought to you by Olink Proteomics. To contact the hosts or for further information, simply email info@olink.com.